Hi there, my name's Graham Gilchrist. I'm a postdoc at Princeton University and GFDL. Thanks for tuning in to my talk. Today I'm going to present some things that I've been working on over the past several months, where I've been trying to understand and discern the timescale or timescales on which the ocean's biological carbon pump impacts the Earth system. This is work that's been done in collaboration with a number of people, including Julius Busecki at Columbia, Sarah Sluniger and Rick Slater here at Princeton, and John Lauderdale at MIT. And my time working on this has been funded through a number of different agencies and projects. So it's well established that at a steady state, the ocean's biological carbon pump, which involves the growth and sinking of organic matter, plays a crucial role in the partitioning of carbon between the atmosphere and the ocean. However, its role in the transient evolution of this partitioning is much less clear, in large part because the intrinsic timescales of the relevant processes are not well known, and we often just cite canonical values for the re-emergence times of water masses at different depths. The major challenge is that the biological carbon pump involves a number of different processes, each with their own associated timescale, from the growth and export of organic carbon from the surface ocean, to its remineralization at depth, and to the circulation that brings dissolved carbon back to the surface. The impact on atmospheric carbon, the integrated timescale of the biological carbon pump, is therefore a complex function of these individual timescales and their spatial and temporal structure. There's been some nice work from the group of Mark Holzer that seeks to better understand and constrain these timescales. Another way of inquiring about the intrinsic timescales of the biological carbon pump is to ask, on what timescale does atmospheric carbon, by which I mean CO2 here, feel ocean biology? Or alternatively, what is the imprint of ocean biological variability on these pictures of atmospheric carbon change, where we might wish to consider geological timescales, the recent past, or perhaps most pressingly, upcoming centuries? What will be the impact on those future projections of changes in the ocean's biological carbon pump? Although the geological past is a very interesting question, I'm here going to focus on decadal to centennial timescales. Our approach to discerning the intrinsic timescales of the biological carbon pump is to think about the response of atmospheric carbon to a pulse in ocean biology. This comes from considering an impulse response, or Green's function, which describes how, given two time series, a target and a forcing, the target can be written as the convolution of the forcing with the impulse response function g. In our case, with the target as atmospheric CO2 and the forcing as a metric of ocean biology, for example the export production, g describes how atmospheric CO2 responds to a pulse in ocean biology. In the schematic, for example, we can see a response involving an initial impact that decays over time. There are crucial assumptions to acknowledge and keep in mind here, which are primarily that, over the timescales of interest, the interaction of the target and forcing is both linear and one-way. That is to say, the target doesn't impact the forcing. As well as being inherently insightful, knowing the impulse response function allows us to extract the imprint of ocean biological variability on atmospheric carbon variability by performing the convolution with the forcing time series, again keeping in mind the aforementioned assumptions. Here I'm showing a purely illustrative example where I've taken two different impulse response functions and convolved them with integrated primary production to derive the component of atmospheric carbon variability, the blue line, that is due to biological variability, the green and red lines. This figure shouldn't be taken literally, as I've not appropriately handled the scaling of the impulse response functions here, but it's simply to show the potential applications here. For the rest of the talk, I'll outline how we have been attempting to evaluate the impulse response function in three complementary ways, including forward perturbation experiments, idealized box models, and statistical analysis of the CMIP archive. First, I'll discuss how we actually simulated a pulse of organic matter in an Earth system model to derive the full, nonlinear forwards in time response of atmospheric carbon. The model we used was GFDL's ESM2M Earth system model, the details of which are shown here. Crucially, we're able to run the model with multiple instances of the biogeochemistry model, meaning that we can induce a distinct biological perturbation while maintaining the exact same model physics and circulation. After a long spin-up, we induced a pulse of organic matter in the euphotic zone by instantaneously doubling the biological growth rate for a single year before returning it back to normal. We also performed the opposite experiment, in which we prevented growth for a year. The perturbation has a number of ramifications, including shifting the nutrient distributions. And to understand the impact of that and to isolate only the impact of organic matter sinking and re-emergence, we ran additional experiments in which we restored nutrients to values from the control simulation. But today I'm just going to present the runs without that nutrient override. On the right, you're going to see an animation of the evolution of anomalies in ocean carbon relative to a control during the perturbation in which the growth rate was doubled. On the top plot is dissolved in organic carbon, and on the bottom is organic carbon, where each is the zonal integral of that carbon pool. So latitude is on the y-axis, on the x-axis. What you will see is the rapid growth of organic carbon in the surface ocean, that's the impulse, which strips out surface inorganic carbon. And indeed, you can already see the impact of this in the first month in the still that's there at the moment. You will then see the organic carbon rapidly sink and remineralize back into inorganic carbon, establishing a persistent vertical gradient in inorganic carbon that is slowly eroded as the circulation brings excess carbon back to the surface and into the atmosphere. I'll just let this run for a time. As the organic carbon is remineralized, the perturbation is eroded after around 12 years in the organic carbon side, but the redistribution of inorganic carbon persists, as you can see here. So what's the impact of that impulse on atmospheric carbon? Plotted here is the evolution of atmospheric carbon over 30 years, atmospheric CO2, over 30 years following the perturbation. This is the impulse response. We can see that, as we would expect, the impulse of organic matter production induces a decrease in atmospheric carbon, which is diminished over time as the organic matter is respired and mixed back to the surface ocean and returned to the atmosphere. Here's the same response for the zero experiment on the bottom, and we can see that they are quite remarkably symmetric, noting that I've flipped the y-axis on the bottom plot here. This gives some hope that the assumptions of the system's linearity might hold. We can confirm that the impulse is entirely due to changes in the atmosphere-ocean exchange by plotting the cumulative integral of the ocean CO2 flux, which falls exactly along the line. Finally, we can show that the decay of the perturbation with time is well approximated by a power law relationship, here plotted in red. Based on these, the approximate half-life of the perturbation is on the order of 5 years, giving an impression for the timescale on which the atmosphere feels ocean biology. Next, we considered whether an impulse response function of a similar form could be found in a highly simplified box model. The model that we used, called Microcosm, was developed by Mick Follows and John Lauderdale at MIT, and it's a very basic 3-box model, but with a comprehensive representation of ocean biology and the carbon cycle. The model was originally developed to consider the global ocean and the deep overturning circulation. However, given the timescales of interest here and the patterns evident in the full ESM perturbation experiment, I adapted the configuration to instead approximate the shallow overturning cells of the equator and subtropics. As for ESM2M, I performed a number of perturbation experiments to discern the impulse response, and with the very short running time of the model, I'm able to explore its sensitivity to the model parameters and configuration. And here I'll just show the sensitivity to the strength of the prescribed overturning circulation. Here I'm showing the impulse response for the perturbation experiments in which we zero out the growth, so kill everything in the surface layer for a year, inducing a pulse of carbon into the atmosphere and a subsequent return to the ocean over time. The shape of the response is broadly consistent with that of the fully coupled Earth system model, and is also well approximated, although less well so than the full Earth system model, by a power law relationship, but with generally shorter perturbation half-lives, generally shorter than 5 years. This is like the result of the smaller domain and absence of any high latitude processes here. The different colours correspond to runs with different strengths of overturning circulation, and the relationship between overturning strength and the exponent of the power law fit is shown in the bottom right. Here, a more negative exponent means a sharper drop in the perturbation. Somewhat counterintuitively, an increase in the overturning strength slows the return of carbon to the atmosphere. This needs further investigation, but I suspect it results from the manner in which the overturning redistributes nutrients between the boxes, and how this is disturbed by the perturbation. Either way, I intend to adapt the model further to give a more thorough treatment of the circulation and the remineralization process. Our final effort to discern the form of the impulse response function does not rely on perturbations at all, but rather seeks to estimate it directly from the statistics of unforced control simulations of Earth system models. Inspiration for this approach came from the work in physical oceanography community, specifically that of Yavor Kostov. Here they use the CMIP archive to derive the relationship between Southern Ocean sea surface temperatures and the southern annular mode, a dominant mode of atmospheric variability, revealing that the impulse involves an interesting two-timescale response. Shown here is actually the step response, which is the cumulative integral of the impulse response in time. Here, the impulse response function was approximated simply using the lagged correlation between the forcing and the target time series. We would like to do the same, deriving a relationship between atmospheric carbon and ocean biology, but there are a few challenges we need to address first. Firstly, atmospheric carbon is strongly impacted by land carbon changes in these models. So instead of using, and also in the real ocean, in the real Earth system. So instead of using that as our target time series, we use the atmosphere-ocean carbon flux. The time series of global integrals of CO2 flux and primary production in GFDL's ESM4 model are shown on the bottom right. Even there, however, the imprint of ocean biology is likely to only be a small part of the total variability, with several other processes potentially impacting the exchange, and this introduces a potential signal-to-noise issue. Consequently, we need to derive an empirical model for the atmosphere-ocean CO2 flux with multiple predictors, so that we can isolate the imprint of the biological carbon pump. Our first step towards this was to consider the impact of surface heat fluxes that, in a spatial sense, are highly correlated with carbon fluxes and known to strongly impact their temporal evolution. Looking first at GFDL's ESM4 model, we find indeed that there is a close correspondence between the integrated heat flux and the carbon flux, raising the possibility that the biological imprint could be found after this impact or this correlation was removed. However, this turned out to be a red herring, since the relatively close correspondence between these variables in ESM4 is an outlier amongst the CMIP archive, as seen here on the right, with none of the other models showing anything close to that linear relationship. The upshot is that a more comprehensive statistical approach is going to be required to confidently isolate the biological signal and estimate the impulse response function across the CMIP archive. In summary then, we have been exploring the intrinsic timescales of the ocean's biological carbon pump by considering the impulse response of atmospheric carbon to ocean biology. The perturbations of a fully coupled Earth system model, we found an impulse response function that was broadly symmetric between the forms of the perturbation, the decay of which was well approximated by a power law. The perturbation was relatively small and its decay was quick, implying that the imprint of ocean biological variability in the atmosphere on decadal timescales is small and short-lived. An idealized model configuration found similar shapes and timescales for the impulse response, but requires further refinement of processes. And finally, determining the impulse response function from control run statistics is ongoing and requires a more robust empirical model. Thank you very much for taking the time to listen. As you can see, many aspects of this work are still in progress, and I would greatly value comments, questions, and critical feedback, so please do reach out. Thank you.